Good morning, ladies and gentlemen. Thank you very much for your time here. I was invited to speak here, so thanks first of all to UBM to have me here as a speaker. The title was set together, and it was quite a complex title: Are there learnings between API, small molecules, and bioprocessing? I'm Lucas Hüdiger. I'm running the North America Pharma Development and Production Service for Pharma Service Group, part of Thermo Fisher. I'm a chemical engineer by training, so I have a lot of history in API, and you will see some of the learnings, or maybe more a bit on the API side than the bio side. I will represent here, for this question, our viewpoint. This means the Thermo Fisher viewpoint. This viewpoint will not be shared by a lot, maybe others, but it's our viewpoint. I think it's worthwhile to share it together. So let's start first maybe introducing Thermo Fisher and the Pharma Service Group. Thermo Fisher, as many of you know, is a large company, very broad field of services. Altogether, we are 70,000 colleagues worldwide, 20 billion in sales per year. And we have one mission in life, and that is to enable our customers to make the world better, healthier, and safer. And I think the CMO space, the pharma space, is really at the core of this mission. As you all know, Patheon, my previous company, was bought by Thermo Fisher a year ago, and we were merged together with the clinical trial division, the Fisher Clinical division of Thermo Fisher. So we now created together the Pharma Services, a fully integrated service group that does everything from API or bio, cell line development, synthesis development, the clinical trial productions, moves forwards to the clinical packaging, labeling, and the clinical logistics. On the other hand, we do everything from large-scale API bioproduction into large-scale OSD and sterile productions. So on the commercial side, we can really cover everything on the development side as well. So for us, the acquisition by Thermo Fisher created a really unique opportunity. First of all, we cover now the full pharma development spectrum with the clinical trial division as part of us. On the other hand, we have a massive backward integration into technologies. Just think about biologics. You use media, you use cell lines, you use gene sequencing, you need mass specs. That's all technologies that Thermo Fisher has, where we now have them within the family, and we are like, the first to use them, the new stuff, give feedback to the equipment development part. So I think it really enables further the development of the service platform that Thermo Fisher has. On the bottom, you see all the different business units in Thermo Fisher, divisions. Starts from specialty diagnostics, analytical instruments, the whole biotech life science solutions, and all the lab tech products that you may know best. The last commercial slide, and I will come into the actual topic. The Pharma Service Group, as I mentioned, was formed out of the clinical trial division, out of Fisher Clinicals and Patheon. We are together at the moment 12,000 people, 2,600 scientists, only focused on CDMO work, pharma service work. We deal with all the technologies already mentioned. We have more than 800 customers, so this shows that we are from a large pharma customer to the really small customers. We cover everything. We really cover the full service breadth. If you start with the chevrons, we can source ingredients, we can develop synthesis, we can develop cell lines for you to get the ingredients. We can design your formulation if it's OSD, if it is soft gels, or if it is sterile. We can scale up all technologies to real full scale. With our new Fisher Clinical friends, we have now access to really develop with you together the clinical trial strategy, the logistics at the end to run all the clinical trials, and then, as already mentioned, the full scope of commercial production. So it's really a nice setup. Large, it has depth, it's a massive technology toolbox, and I think it can actually solve most of the challenges that you have in your pharma development area or the pharma production area. So what is the focus of the talk today? I was asked to brainstorm if there are learnings between small molecule, I will use the term API, and biologics. or the processing side. As you see from the breadth and depth what we have in the Pharma Service Group, we have all the technologies and we have all the depth. So I will represent our viewpoint here, what we see, how we see this, especially focusing on the drug substance part. I think the learnings are more on the drug substance than the drug formulation, but we'll cover some of the drug formulation part as well. And please keep in mind, these are our ideas. They're not, the industry ideas, or maybe not have a broad consensus, but these are our viewpoints. Let's start first with a historic viewpoint. We all know small molecules. We all occasionally use them, if it's an ibuprofen and aspirin. When you look at the history behind it, aspirin started in 1899, so it's very old. So the small molecule processing is a lot of history, a lot of experience, a lot of development since then. When you look at the bio side, the first bio product was humulene, 1982. It's a relative new technology, especially in the pharma world, of the speed that the pharma world is moving. So I think there's a massive difference on the maturity of the processing that both technologies are using, but also by the technology itself. So I will go more into these details as well on the maturity side. When you look at the NDA approvals, you see that small molecules have a constant demand. If you would actually look back to the late 90s, early 2000s, you would see more or less a similar number of new chemical entities. Depending on the year, they fluctuate a bit. I think what changed in the last couple of years is that the blockbusters that we've saw in the late 90s, early 2000s, they're now moving out. We have many more orphan drugs that come to market. On the bio side, you see a constant growth. But that is important. There's a constant growth on the bio side, but small molecules are still here, and I think they'll stay here. When we look at the current pipeline, or the pipeline data here from 2017, a very similar picture. We have still the majority of the molecules are small molecules, so APIs. But when you look back historically, the biologics part increased tremendously. And we should also not forget, when you look into generics, a couple of years ago, there were no biosimilars on the market. Now we have biosimilars on the market. So from that point of view, biologics, they're getting more mature as well, but there's still a massive focus on small molecules. When we look at the processing, what we'll talk will be mainly the actual drug substance processing part, from cell line development over the pre-cell cultures. The actual cell culture here, now we can use stainless steel or single use. Cell purification at the end, viral removal, and then the drug formulation, the bulk drug formulation. For us, bulk drug formulation is an important step that actually then dictates how easy you can formulate a drug in the final vial or prefilled syringe. So I will mainly focus on the first steps. The sterile filling, it is a complex operation, but when you look at unit operations, it's compounding, it's sterile fill, it's filtration and then filling. So from that point of view, it is not so complex. To run it properly, GMP-wise, that's a different story. When we look at the API part, here we have two important buckets. One is on the chemical synthesis part. You have several steps, and I think that's one of the major differences, where in the cell line you have the cells, and then you actually from the cell you grow down to the whole process. On the chemistry side, you design your synthesis path, and depending how you design it, you have several starting materials. The schematic part here, it's A and D, and then you use multi-purpose reactor trains. Some of them are looking very similar. You always have a crystallization with small molecules. Most often, I would say 99% are solids. You may have special equipment like for hydrogenation reactions, special materials for very acidic or very caustic reaction systems. But at the end, it's always a reaction, a quenching, a crystallization, and then you have an isolation part. You then move over to the OSD side, which is normally blending, granulation, drying, and then compression. Here again, we mainly focus on the API drug substance production and not on the rest of the actual drug product part. You know, there's a big question if there's actually learnings, and I think here one big difference when you look at the whole universe is the size of the topic of the drug substance that we talk about. You have aspirin on the left-hand side. You see the size is the indulgence. You see aspirin with 180. You have monoclonal antibodies, some of them up to 150,000 daltons. So they're almost a factor 100,000 larger than what we have on the small molecule side. So I think there's a massive difference in processing. There's a massive difference the way how we actually analyze. On the other hand, when even we focus on APIs, APIs are not just APIs. You have simple APIs like aspirin, salicylic acid. You have very, very complex structures in some of the oncology products with a lot of stereocentric centers, very complex reactions. Some syntheses have up to 20 steps. So I think there's a difference within the category as well. But I think important is there's a massive difference. And even if you talk about bio, it's already a question, do you actually target insulin, which is a microbial fermented product, or you really talk about antibodies or fusion proteins. So just keep this in mind when we go through the learnings. I'll now first focus on a couple of API learnings for the bio sector. All of us who worked years and years now in the API field, we all got used to the price pressure we have, I would say, since maybe the late 90s. There was, for quite a while, an overcapacity in the markets, a massive price pressure, end of blockbusters, as a price pressure on the pharma side. And I think what we learned was a real good development on the synthesis path. And this already starts in phase 1, phase 2. You actually put a lot of effort in to create the right synthesis path. But then when you actually look what happens in phase 3 till you go into the generic phase, you actually do a massive amount of additional work. And the work you do in several areas. On the one hand, you try to optimize the processes. So you take the existing process, you optimize, you try to reduce starting material costs, you increase yields, you reduce waste. Solvent recycling activities like this, which has a significant impact on the costs. On the other hand, normally in late phase 3, just after launch, most companies actually review the synthesis path and see if you can actually create a much cheaper synthesis path for the later commercial phase. And this results in a massive cost reduction. And instead of quoting numbers, I took factors. So if you have a generic a couple of years after the generic launch, if you give this a factor 1, then you have normally 5 years after launch, your drug substance price is 2 to 3 times higher. At launch, it's 3 to 5 times higher. And then you see up to phase 1, you talk about the 30 to 40 times higher cost base. What is the cost base? Here, it really depends on complexity. It could be on the generic phase, it could be a couple of $20, $30, $50 per kilogram. It could still be in the thousands of dollars, depending on the volumes and the complexity. On the bio side, there's a difference. First of all, when you look at the cost reduction between phase 3 and the generic biosimilar phase, there's only a factor 3. Reason here is that the process dictates the end quality. This means that you cannot change the synthesis path. To get a new cell line, you get higher titers, but you're not getting 2 or 3 times higher titers or processing costs. I mean, that's a really, really important change. On the other hand as well, since the process designs the quality of the product, there's not that much difference from early phase to late phase, because whenever you go into phase 3 PPQ work, you're at scale and you already defined clearly what media to take, and there's no changes later on in the process. From that point of view, I think from a learning perspective, if Bayer can learn something out of API, have a real good study of the cost, learn how to optimize the cost, and I think the biosimilar trend will really catalyze this. Have a close look at further cell line development, have a look how we can actually reduce cost on the media side, single-use side, and also then on especially in the downstream processing side. So that's one of the learnings. When you look at history as well, you see on the equipment side, You know, the API business now, fine chemicals, you know, it's an industry that is more than 150 years old. You see here on the left-hand side, it's an old hand centrifuge that was used the late 1890s to separate solids from a mother liquor. You see the right-hand side, a modern centrifuge. Here you see how much development actually went in the last more than 100 years on the equipment development side. Noble centrifuges, they have very thin layers, so you can actually isolate very fine crystals. You can run a lot of different wash cycles. They're self-loading, unloading. So I think that was a massive change, and I think it's a good development. Learnings here for the bio side, and I will come back more to the bioprocessing side, but I think the key learnings. The bioprocessing part depends a lot on single-use equipment. If it's single-use bags, single-use reactors, a lot of filters. Here, I think every company, every customer tries to standardize their own stuff, and there's not large industry standards. I think here, simplification of the technology, standardization of filters, bags, single-use reactors will really help to drive the cost down. Media standardization will help. You know, the more mass produce you can get media done, the cheaper it will get. I think there's a massive learning here that we can really focus. Downstream processing was a big move on the chemical side. You know, crystallization is one of the timely actions that we have in the synthesis path. Here as well, you know, when you look at the downstream processing on the bio side, all the filters, all the column separation that you do here, if you can move forward with crystallization, protein crystallization, I think there's a massive simplification and speed that you would gain. Next learning from the API side, I think on the API, in the API world, we have a unique circumstance that we have all the analytical technologies available to actually fully characterize a molecule. You have mass spec that can easily go up to several thousand daltons. You have all the UPSCs, HPLC technologies, you have gas chromatographs. We have, on the other hand, electron microscopy, mulverns, you can measure surface areas, so you can fully characterize, if you spend enough time, the full API. So you have, at the end, two controls of the quality. On the one hand, you have the process that actually defines the quality. On the other hand, you can, after the production, you can actually fully analyze, and you can absolutely be sure what you have produced is what you actually want to have. This is different on the bio side. On the bio side, unfortunately, still now, the process dictates the quality what we have at the end. And I think further advantages here, if you could further develop complete protein characterization, I think this would really bring a big help and would enable further process improvements on the bio side. Fortunately, I think several companies move forward, especially in the mass spec area. Thermo Fisher is one of them who really now delivers the tools to further fully characterize proteins. I think that's a big step forward and will really further develop the whole process in years to come. Another learning is building blocks. When you look back at chemical synthesis, chemistry was based on fine chemicals. You know, if you start to do an API synthesis, you're not starting to write your own phenol or your own benzene ring or whatever, you buy them in. You buy in starting materials which are already fine chemicals, maybe with one or two more reactions on top of it. So we got used in the API world to really use building blocks. There are other areas when you look at peptide chemistry, where you have the amino acids, you have your solid phase reactions, you assemble longer chain fragments, then you couple them, and at the end, you couple the fragments and you protect and you analyze the final peptide. So I think this area, you know, it really is building block technology. In bio, we don't have anything like this. We have fabs, and I think they're here, you know, maybe there's a radical potential in the future to mass produce part of the proteins and then add and dock the active proteins that you really want to do, so that you have actually skeletons mass produced and then really tailor-made your warheads to this. I think conjugates, drug conjugates already do this. We already mentioned the full characterization of APIs. Here, I think besides the chemistry, the solid chemistry is really important as well. And we all know that whoever works in the OSD side or solid dose side, APIs always creating issues, even if the synthesis is stable. Color slightly changes, particle size slightly changes, and this may have a massive impact on the solubility at the end. On the API side, we have all the technologies to measure it. We not always use it, but I think that the measurements are there. We can isolate polymorphs the right way. You have all the Malvern, the density analysis. We can actually really well characterize the flows. I mean, that is all working. I think here on the bio side, three-dimensional protein analysis will have a massive impact on the formulation side as well. So here further developments, learnings that you have to characterize not just the chemistry part, but also the three-dimensional structures. This will create a massive further processing improvement in the future. Main benefit would be, especially when you look at formulations, if you can really use the holding time properly, the yields will go up, which has a massive impact on your cost base. Now let's focus on the bio learnings for API. Here, I think a couple of just initial slides on technologies. I think bio uses a lot of in-process controls, a lot of dedicated sensors, a lot of software. All the parameters are actually controlled, analyzed, and actually utilized because the process dictates the quality. Bio is also in some areas already further advanced in continuous manufacturing, perfusion technology on the drug substance side. The sterile fill is more or less a continuous side as well. Parameter analysis studies really create a stable process. You see here some diagrams of initial trial runs, and after parameter optimization and good studies with inline sensors, you can actually really get stable curves. And this all enables us to really start more or less to run continuously from a media, perfusion, and then go into fill finish. Learnings here for API is that we do everything at the moment in API more or less the batch way. There are microreactors, the loop reactors. The pictures here on the left side show some microreactors in the Linz Pharma Service Group's division. We have full setups from pilot into full commercial. We can run continuous, but this is a niche technology that we only occasionally use. When you look at OSD capacity, capabilities, there's a big trend at the moment to go continuous. You see in the right-hand side pictures of our continuous manufacturing suite in Greenville, North Carolina. Technology is relatively simple on paper. You have mass flow controllers, and then you either do continuous granulation, drying, or continuous capsule filling compression. Sounds easy, but the big issue is solid characterization. If you want to dose a solid absolutely accurate, you really have to understand this. But here, learning for API is get more data during your processing, run more continuous, drive further developments like the continuous side. I think this will help us in the future to further cut the costs here as well. Process qualification and validation is another key topic. We all heard about CPV, continuous process validation. On the bio side, you have to analyze this data much more accurately because the process dictates the quality. On the chemical side, we do this half-heartedly. I think here there's a massive development that we really bring into the API side as well. Analyze batch data, really control the manufacturing data with online sensors, bring a much more stable process together so you have less variation. And this will really help us at the end to increase yields, to decrease byproducts, and at the end, really be able then to go fully continuous in the future. Last learning from the bio side. Bio spent an enormous amount of time analyzing raw materials. This starts from the cell line, goes into the resins that you use for the single-use bags. There's a lot of leachable work done on the single-use bags, on the vials that you have at the end for the sterile fill finish. So there's an enormous amount of work done in characterizing the ingredients that you actually use for the process. Key reason again is whatever you add is at the end dictates what quality you have on your product. On the API side, I think there's some learnings for us. Since we have this strategy to have different starting materials, intermediates, and we assemble an API at the end, we really need to control, we have a chance to control better intermediates. And you should not forget, when you have a starting material, normally you define it to 0.1%. every impurity below 0.1%, you know, occasionally you don't even qualify exactly what it is. This creates, even in generics, occasionally issues that you have suddenly different colors, a new byproduct, because somewhere up front someone did a change in the process and he did not really see what was done. So here the learning for API clearly is the more time we would spend on analyzing, the more data we would actually use to actually control the process, the more stable we even get on the API side. So let's come now to the final conclusion. I think we all in the pharma area, in the pharma service area, we have one goal in life, and we really want to address the unmet needs of our patients. I think that's our goal in life. We all know there's a lot of unmet needs. There's a lot of still, you know, a lot of orphan drugs need development, a lot of cancers need treatment. I think long term, both technologies, if it's API or bio, they have a real a key role in life. We need the small molecules. They have open access to drugs around the world. From a cost perspective, they're cheaper. I think here we can address some of these increased healthcare costs. On the other hand, with the bio in a completely different maturity pace, I think we need bio drugs to especially to fight cancer, to move into gene therapy and completely novel treatment forms. So both technologies will be needed. Cost and waste reduction will be key for both. I think I showed you that both parties can learn from each other, but I think it's really important. I think in-process control, continuous process validation are some of the key areas that we both have to focus on. Seamless integration of drug product and drug substance manufacturing will help us as well. Drug substance dedicates how fast we can move on a drug product. And I think here, the more we integrate the service, the development, and then the production, the leaner we can actually do this, and the more cost efficient we can do this. The technologies will move on. I maybe highlighted building blocks as maybe a bit a crazy idea at the moment, but I'm sure, I think the bio universe will come up with their own answers to cut costs, to increase the speed on the development side. I think there will be novel breakthrough technologies coming. It will not just be cell therapy, viral therapy, or gene therapy. I think the bio side, I think there's a lot of room as well for improvement. And then the last point for us, really important, integrated supply chains will be the key topic for the future. And here it is quality and process control that really dictates how lean we can actually run this. Jim Mullen before, you know, he mentioned Amazon and Apple. I think if you really want to go down to those models, you know, we on the supply chain side, we have really to make big areas. And I think here, both sides can learn from each other, both have still a lot of work to do, but I think there's a good chance that we can really get leaner costs all together and better quality at the end, if you really learn from each other and move forward. With this, I maybe have time for one or two questions. Thank you.